In this video, we will demonstrate how you will conduct the Salmonella detection in the lab. You will find the explanation on the principle of those media and what the positive reactions look like in the next video. On day 1, you will evenly sample 25 grams of food to 225 ml of buffered peptone water. Homogenize for 2 minutes. The stomach bag will be incubated at 37 degrees Celsius for 24 hours. The following day, you will add 1 ml of the pre-enrichment to 10 ml of the tetrathionate broth or TT and 0.1 ml to Rappaport Vasiliades or RV. Both tubes will be incubated for 24 hours. However, TT will be incubated at 37 degrees Celsius while RV at 42 degrees Celsius. On the third day, to continue on the conventional method, you will vortex and streak selective enrichment broth onto bismuth sulfide agar and xylose-lysine deoxycholate agar respectively. First, you will split the two types of the agar plates into half, and on each half, you will label which selective enrichment it comes from. Dip the sterile loop into TT broth and streak onto the halves of the plates labeled as TT. Because the first phase usually contains a lot of liquid after streaking, make sure you dry the plates under the Bunsen burner before you start to streak the other halves of the plates. In that case, the liquid from the first phase will not flow around to contaminate the entire plate. Continue the same technique for RVing. The plates will be incubated at 37 degrees Celsius for 24 hours. On the same day, you will also do the rapid detection using RV and Reveal 2.0 Salmonella test device. You will add 500 microliter of RV to a 1.5 ml centrifuge tube. Notice the reagent zone on the test device. Submerge the reagent zone of the test device into the RV in the centrifuge tube to allow reaction to happen. You will observe the result after 15 minutes. On day 4, you will observe selective and differential agar plates and circle out one presumptive colony from each half of the plate. The idea is that isolates coming from different medium combination are more likely to be different strains, and your confirmation tests should include as diverse isolates as possible. The 4 circled presumptive colonies will be streaked onto 2 divided triptych soil agar plates or TSA. TSA will be incubated at 37 degrees Celsius for 24 hours. On day 5, you will pick one single colony from each half of the TSA plate and use the same colony to finish all the confirmation tests. The confirmation tests include 2 biochemical tests and 1 serological test. To conduct the biochemical tests for each colony, you will have triple sugar iron slant agar or TSI and lysine iron slant agar or LIA. Label the tubes indicating which colony you will test, and then you will streak the colony onto the slant of the agar in a zigzag way and stab the agar to around 1 to 2 centimeters from the bottom. Use the same technique to finish the tests for the rest of the colonies. The slant agar will be incubated at 37 degrees Celsius for 24 hours. For the serological test, you will need the reagent Salmonella O antiserum poly A2I and VI and a microscope slide. Separate the slide into 4 sections and label each section to indicate which colony you will test. Add a drop of the reagent onto the slide. Use a sterile loop to touch the colony on the agar plate and mix with reagent on the microscope slide. Rotate the slide for 1 minute and observe the result. Reaction that happened beyond 1 minute is not considered. Finish the test with the rest of the colonies. For each slide, you can conduct the serological tests for multiple colonies. One slide should be enough for testing 4 isolates, but if you feel uncomfortable with the limited space, you could use 2 slides. On the last day, you will observe the results from the biochemical tests. We will discuss how to annotate the result in the next video.